All right, so I need you to focus on neurophysiology, and I know that's hard for the majority of you that haven't taken your muscle test yet. Some of what we talk about will ring a bell because it applies to both nerve and muscle, but most of it not. It will still kind of make a foundational structure that may make the muscle physiology make more sense as we get into it. At least it potentially does. And I called this review from lab. I just added this morning, but it's not a review from lab because y'all haven't done this lab yet. So we're going to go over these very, very briefly. And then when you get to lab tomorrow, you'll already know it. So it'll help at some point. So you know that the neurons are the functional cells of the nervous system. They're the ones that initiate and conduct nerve impulses and give you all your nervous system activity. The classes of neurons are divided into two kinds, and the first kind is the structural class, and the structural classes are based on the number of cell processes. Now, the word process is an unfortunate one because it has so many different meanings. I'm not talking about physiological processes here because this is a structural category. So when a cell has a process, what that means is it has things sticking off of it. Just like a bony process is something that sticks off. Okay, so a cell process in the structural sense is like a bony process. Are you with me on that idea? So cells, neurons, can have a bunch of processes. This particular one's kind of star-shaped, so it has 5 processes, okay? Some neurons have only 2 processes, one kind of at each end. And some neurons have only 1 process that runs in both directions. So it's sort of 2 parts, but technically it's 1 process. Can everybody see my 3 categories? The one that has only 1 is called a unipolar neuron. Each process forms a pole. Okay, so a unipolar neuron has only 1. Sometimes these are called pseudo-unipolar. What does pseudo mean? False. And why would it be called pseudo-unipolar? What did I just tell you about that one process? Kind of has 2 parts. So it really has 2 parts, but they fuse together before they attach. So as it attaches to the cell body, it looks like 1 part, but all neurons have to send signals in 2 different directions. Some signals have to come into the neuron and some have to go out, so you need 2 parts. Okay? So whether it's called pseudo-unipolar or unipolar, it's the same thing. So what do we want to call this guy in the middle that has one that the impulses are coming in and one that the impulses are going out? What do you want to call that one? That's a bipolar neuron. Has nothing to do with the category of bipolar disturbance of the brain. Has nothing to do with that. All right, so anything more than that, the number is variable. It can be just a few or it can be a lot. So what do we want to call something that has more than 2? Multipolar, because we're biologists, we do not like to count higher than 2. So we call this multipolar. Now, I'm going to tell you there aren't very many places in the body where there are bipolar neurons, and you're not going to study that one in lab. I'm going to go ahead and tell you where they are. It's in the notes anyway, but I'll go ahead and tell you. Bipolar neurons are found in the retina of the eye, where they form a whole layer called the bipolar neuron layer. And they're found in your olfactory sense, which is in the roof of your nasal cavity. That's the only place I know of in humans that have bipolar neurons. In both cases, they are sensory neurons for sense of smell and for sense of vision. Are you with me on that? Okay, that's the bipolars. We'll see for our others, the functional classes are going to be correlated. So our unipolars are all sensory. Every one of them is sensory. Okay? So if I'm talking about your tactile sense, what was your tactile sense? Sense of touch, thermoreceptors, pain receptors, stretch receptors in your gut, whatever. If it's a sensory function, then it's unipolar with two exceptions. What were the two exceptions? I just gave you two places that were bipolar. Right? Olfactory and one of the layers in the retina of the eye is bipolar. And every other sensory neuron is unipolar. Okay? So what does that make every other neuron in your body? Multipolar. So multipolar is the most abundant of the categories. All right? So far? Okay, well, let's look at our functional classes. Our functional classes are based on which way that neuron is sending an impulse. So our functional classes are sensory and motor. Leave a space in between them because there's a third kind, and our third kind is called integrating. So sensory neurons are also called afferent. What does the word afferent tell you? It's carrying something toward a destination, toward, in this case, a center. Okay? So what's the opposite of afferent? Efferent or efferent, however you want to pronounce it. Some people say afferent and efferent just so they can remember how to spell them. That's fine. Most of us that have been doing this a while call it afferent and efferent. Okay? So our integrating guys are also called association. Gee, I wonder what they do. They connect other neurons to each other. They integrate the information coming from other neurons. All right, so let's try to relate these to those structural classes. What functional class do our two examples of bipolar neurons fit in? Afferent, or what was the other term? It's right up there on the screen. Sensory, same thing. Okay, just start associating those terms. And so what about the unipolars? Unipolars? Sensory. So our sensory guys are all unipolar except for those two little places I gave you that were bipolar. Okay? And then what about my integrating guys? What are they? They must be multipolar. And what about our motor guys? Those likewise are multipolar. Okay? So if I ask you what's the most abundant structural class, your answer is multipolar, because your brain is almost entirely made of association neurons. And those are all multipolar. They take different shapes. We have different names for different kinds of them. We're not going to get that bogged down in it. We're just going to call them multipolar and recognize them when we see them. Okay, so far? Okay, so that's not a review from lab. That's a review, really, for lab. And that wasn't in the handout anyway. All right, so what we're going to look at next is something on the neurophysiology. And here I want us to examine this question, and you're going to work in your groups on this and just come up with what you think the attributes of a battery are. And I've made a little circuit here. So the first step is to determine what are the parts of the circuit that you see. So work in your groups, and you should be able to list at least 3 obvious parts of that circuit. Okay, so what three parts do we have? What do you see in the picture? I see a battery. Very good. Good job. That's exactly what I'm asking you to do. Just list what you see. That's the first step. And what else do you see? I see a wire. I drew that myself. Can you tell? What's the third part that you see? A bulb. You see a bulb? Very good. Okay, so what makes the light bulb light up? Do we understand how electricity works? How does a battery work? Does anybody know how a battery works? Not really? You need to know how a battery works. I don't know as much about it as I probably should or could, but I know a little bit about it. I know enough about it to teach you what I think you need to know, which is that if you looked at the inside of a battery, what you would see internally is there's a compartment that completely seals off our two sides. Okay? And the two sides have solutions in them with opposite electrical properties. And those opposite electrical properties result in having a lot of electrons on one side and a deficit of electrons on the other side. Okay? You don't need to know what the solutions are. One of them's acidic, though. You knew they're battery acid. You've heard of battery acid, and this is why we don't play with batteries, nor do we let our Labrador retrievers chew them up, even though they want to. Okay, so how do I get electrons from this side to this side? How do I do that? That's what the wire does for us. So I have an electrode here. This is just a little plate electrode. And here we have a post electrode. And so I take my wire and I run it from one side to the other side. And so now, because this wire conducts electricity, I can just imagine my little happy electrodes as they race from one side to the other side. And when do they stop racing? Battery's going to run down eventually, right? What do you think is going to make it run down? I have a finite number of electrons over here and a finite number of positive charges over here. This works a lot like diffusion. It's not a diffusion gradient. It's an electrical gradient, though. There's a difference in electrical charge. And things will move as long as there's a gradient. So as soon as the electrons from this side have all counteracted the positive charges on the other side, there's no attraction anymore, and the gradient is gone. Does that make sense? Are you with me on that? So what does a battery charger do? It reverses that. It does. You send electrical current into a rechargeable battery, and it literally pushes the electrons back the opposite way. It takes more electricity to do that than you can release from the battery because you have to push them against their will. They really would rather stay with all those positive charges. Does that make sense so far? Okay, good. So how do I put this to work? Here sits my battery, and I have this electrical wire. That's fine. So as long as I've completed the circuit, then the electrons can flow from the negative end to the positive end. But what I really want it to do is to light up this light bulb. So how do I connect the light bulb to the circuit that I made? All I have to do is kind of wrap the wire around it. So I can insert my light bulb in here and make a contact between them. Just wrap the wire a little bit around it so I have a good tight connection, and then my light bulb will light up. Yay, success. The electrical current that's going through that wire, some of that electricity is transmitted to the bulb, and in the bulb, on the inside of the bulb, there's a vacuum, you know, and in that vacuum there's a filament, and that electrical current creates resistance in that filament, and that filament gets hot. And when it gets hot, it glows with heat. So you convert some of the heat energy to light energy, and that's what makes a light bulb work. Make sense? Questions? Okay. We have a missing piece here. What do we need? Do you really want to do this every time you need light? Sit there, take your light bulb, your battery, your wire. What do I need? I need a switch. So what I really need is to be able to break that circuit when I want to, and then when I'm ready, just push the wire closed, and that completes the circuit. And so that's my little on-off switch. So if you are looking at a flashlight, what do you got in a flashlight? You have some pieces you don't need. You have a housing so you don't have to hold the batteries. You stick your batteries in there, and then you got a little switch and a bulb, and then you have a reflector, which is nice but not necessary. Okay, so now you know how batteries work. Isn't that the coolest thing? All right, very good. But the question was, how is a neuron like a battery? Okay, so what do we need our neurons to be able to do? What did our battery do for us? It stored electrical current in the form of an electrical gradient. It allowed that current to flow when I turned on a switch, and I could use it to do work. Are you with me on all that? Okay, so I need to be able to create an electrical gradient. I need to be able to turn the switch on when I'm ready, and I have to hook that to, in some way or another, do some work. Okay? And that's the way a neuron is going to be like a battery. All right, so to make our electrical gradient, we're going to do this first. So we're going to charge our battery. And in our electrical gradient, we're going to find that we have poles. Doesn't our battery have poles? Has a positive pole at one end and a negative pole at the other end. Our cells don't have ends in the same sense that a battery does, but our cells, any cell, whatever the shape, you can look at a round cell or you can look at a neuron or whatever, doesn't matter what the cell is, every cell has, for its entire membrane, three-dimensionally, the entire membrane has an outer face and an inner face. And the outer face is going to be positively charged, and our interface is going to be negatively charged. So that's how a cell membrane is like a battery. If we look at an extreme close-up of just a cell membrane, so if I look up at a close-up of it, I've got my phospholipid bilayer. If this is my extracellular fluid here and my intracellular fluid here, and of course I got the rest of my membrane, I'm just tired of drawing it, then I'm going to have positively charged ions on the outside and negatively charged ions on the inside. Okay? Now, it's not really that simple. There is always a mix of positive and negative. So a more accurate way to say this is to say that really there's just more positives outside than there are inside. If you look at the number of ions on the outside of the membrane, there's just more there. And that makes, and because there are more positives on the outside than there are on the inside, it makes the outside positive relative to the inside. Okay? So if I took a device that would let me measure the outside of my cell and the inside of my cell, here's my little device. It's just a voltmeter. Voltmeters can measure electrical charge. A voltmeter is what you would use to see what your battery charge is. And they can be very simple or they can be very complicated. They actually pack them with batteries these days. You have a little thing, a little card in there that will let you know if your battery's still good or not. Tells you what the charge is left on your batteries. I love those things. I always save them because I have dead batteries all over my house. Nobody will throw batteries away because they might not really be that bad yet, right? So keep a voltmeter handy. A voltmeter has a gauge on it with numbers that will display and some kind of a little needle on it that hooks up to these two wires. And my two wires in the case of a voltmeter that are measuring the voltage of a cell are obviously microscopically small. One of them is going to be on the outside, and it can just be a flat electrode. We just call it a plate electrode. And the other one actually has to punch through to the inside, so that's called a spike or a needle electrode. Okay, so it just punches just barely into the cell. But as soon as you do that, haven't I created a circuit here? Because inside this box, these two electrodes are connected to each other, but they're connected through that needle. So if there's any electrical charge at all, that needle's going to move. And because of the way I calibrated the needle, when my cell is in good shape, then that needle is going to move to the left, which is the negative end. And that's the way I calibrated, because the inside is negative relative to the extracellular fluid. So we say the cell itself is negative. Does that make sense so far? Questions about that? You can actually do this in, if you take a junior level physiology, you'll probably get to do this in real life. And we use all kinds of things. The first experiments that were done with squids. And you might wonder why, but the answer is people dissecting squids found out that their neurons are really big. So they have these neurons that go out into their tentacles. They're big, giant multipolar motor axons that make the tentacles contract, the muscles of the tentacles contract. And they're really big and really long, so you can do a lot of neurophysiology on them very easily. All right, so what we find is when we hook up a voltmeter to a typical neuron, that the voltage is 70 millivolts. And because it is negative on the inside, we say that the charge of the cell is negative 70 millivolts, MV, millivolts. And that's pretty typical for your average neuron. The highest, the most powerful charge I've ever read of is negative 200 volts. That's those giant squid axons. And all of your cells, all the cells in your body are positive on the outside, negative on the inside, because your sodium-potassium pumps are running all the time. But an epithelial cell or a connective tissue cell might be negative 30. So too weak to be a very good battery. Okay? But all your cells have this electrical difference. It just has to be strong enough to do work, and the only cells that have enough of a charge to do sufficient amounts of work are the three cells that we call excitable. Nerve cells, muscle cells, which can contract, and gland cells, which can secrete. Are you with me on the big picture? Okay, so we've got to figure out how it is that cells maintain this electrical charge. They're basically constantly recharging their battery because they need to stay positive on the outside and negative on the inside, even if you're running the battery and doing some work. You don't want your batteries to run down. So we're going to look at the distribution of ions, and we're also going to look at the channels and carriers that give us this setup. And you're going to have a picture in your textbook that's going to show you this. But what I want you to do first is to draw a little sketch. So this is just a cross-section through a neuron. Could be one of the cell processes of the neuron, doesn't matter. And I want you to draw this picture with a big potassium on the inside, a big sodium on the outside, and a smaller, but still pretty sizable, calcium on the outside. Okay, and then we're going to add the anions. Remember, cations have a positive charge, anions have a negative charge. So our anions on the inside tend to be proteins and phosphate. Proteins and phosphate. And on the outside, we have a lot of chloride because chloride and sodium are just in love. Okay, for our anions on the inside, watch as I erase these, because I'm going to replace them with a symbol that we use. We just use a big A for anion. But I want you to remember what those anions were. What were they? Proteins and phosphate. So let's think about that for a minute. What do I need phosphate in my cells for? Phosphate, remember, is the P of ATP. Do I need a lot of ATP in my cells? Yes, I do. So I hang on to that phosphate on the inside of the cell, and I tend not to let it leak out. I would rather have phosphate coming in than going out. Phosphate does not leak out easily. Okay, so far? All right, proteins on the inside of the cell, they're doing something. They may be structural, they may be enzymes, they may be internal transporters, they may be regulatory. Lots of proteins on the inside of a cell. And if you didn't already know this, then I want you to picture it from now on. There are not a lot of proteins in extracellular fluid. I'm going to expect you to remember that in AP2. So what's the deal with extracellular fluid? Hey, it doesn't have a lot of protein in it. Proteins in general are anionic. That helps them repel each other so that they don't all glom up together. There are exceptions to that. For example, collagen we know is a protein that's found in extracellular fluid. But if it is an anionic protein that's just kind of floating around, chances are it's on the inside of the cell, not the outside, and that means we don't let it leak out. So tell me about these anions that I have abbreviated with a big A. They're inside the cell, they're trapped there, they're never going to leave. You simply do not let them leave the cell. They have jobs to do inside the cell. They have nothing to do with the membrane potential, but they're there, and they're not leaving. They're not going anywhere. Okay, so if that's your fixed point that you're starting with, then what would make potassium want to stay inside the cell? Potassium's positively charged, all those anions are negative. Those anions attract and hold the potassium inside the cell. Okay? Doesn't sodium want to also come in? Yes, it does. It sure does. Okay? Doesn't calcium want to come in? Yep, yes it does. So what's preventing our cations from all leaving the extracellular fluid and running into the intracellular fluid? Among other things, I have anions in the extracellular fluid. And chief among these is chloride. Okay? So far? Questions? Okay. All right, so how did I get there? How did I get to where I am? Well, I have channels and carriers. And let's see what we remember about these. So I need some leak channels, I need an exchange pump, and then I need some gated channels. Let's see where we are with that. All right, so if I have my favorite exchange pump, what is that? Sodium-potassium pump. Now, I told you last week not to forget it. So tell me what you know. Thank you for being brave, though. What is it? Backwards. No, wrong backwards. Draw a picture. Put it in your notes. 3 sodiums go out. 2 potassiums come in. Now, if you're feeling bad about getting that backwards, studies have shown that when you make a statement, whether it's verbally in class or on a test, once you miss it, you tend to remember it better. Okay? So, do we all have the answer to this question, what does the sodium-potassium pump do? 3 sodiums go out, 2 potassiums come in. 3 out, 2 in, sodium more than potassium. Got it? So when I ask you next week, what was that again? You're all going to get it right, right? And before you guys that speak up feel bad about it, just remember that everybody else that didn't speak up, yeah, they wouldn't even try. Okay. So 3 sodiums go out, 2 potassiums come in. Can you see that that's how we get to the point where we have lots of potassium on the inside and lots of sodium on the outside? You started this way. Your life started as a fertilized egg, and that fertilized egg came from a precursor cell in your mother's ovary, which already was this way. And how did that cell in her ovary get that way? Well, it was the product of cell division, and the precursor cell already had this. So how far back do you want to take that? Back to the very first fertilized egg cell ever in existence. It was already this way. Okay? This is the way of all cells. All cells default to this. Are you with me on that idea? At least in terms of animal cells, this is the way we work. Okay, so how many of these pumps do I need? If I have a lot of voltage, 70 millivolts is a lot of voltage, then does it make sense that I have an abundance of these little sodium-potassium pumps? What are they all doing? Pretty much all the time. Whether you're awake or asleep, whether you're sending a nerve impulse or not, they're pumping away. They just pump around the clock. And the more of them you have, the more sodium you can kick out, and the more potassium you can bring in, and the more difference you can have between the concentration on the outside and the concentration on the inside. And the greater that difference, the greater the electrical charge, the more millivolts. Okay? So does everybody link the millivolt charge on the membrane with the number of sodium-potassium pumps? When we add on top of that we had these proteins here, right? Proteins that are negative, phosphate that's negative, all these anions. that just makes the inside even more negative. Yes, there's a lot of potassium in there, but it tends to be neutralized by the high number of anions on the inside. Do I have lots of anions on the outside? Yeah, I do. But I still am continually kicking out more and more sodium, and I'm not messing with the chlorides at all. So over time, we get a little bit ahead on the electrical charges, making the outside more positive than the inside. Okay? So that's the job of the pump. What does the pump do? It kicks out 3 sodiums, it brings in 2 potassiums in exchange, and of course it uses an ATP in that process, but because the pumps are on all the time, they are continually maintaining this difference in electrical charge that leaves us with high potassium on the inside, high sodium on the outside. For now, we're going to ignore the chloride, although we're not always going to ignore it. And we're done talking about the anions. Unless something happens to screw up that cell, and in that case it's probably going to die, those anions are just going to sit there and pull positive charges in. Are you with me? Okay. So what would happen if I put a leak channel that lets potassium leak out? Remember, a leak channel just generally is open all the time. It doesn't have much of anything that you would call a gate. So there's my leak channel. And it just lets potassium leak out. What do you think is going to happen after potassium leaks out? What? No, this channel is just for potassium. So what will that do to the charge? It was already positive on the outside and negative on the inside, right? So what's that going to do to my charge? Isn't it going to make it more positive on the outside? Okay, so does potassium leak out easily? Can you let more positives out if it's already positive on the outside? Positive charges repel each other. So potassium does leak out some, but it's kind of like it's trying to leak out against a law of physics, that it's not attracted to the outside. Are you with me on that idea? Okay, but some potassium does leak out. Not a whole heck of a lot, but some does. We can measure that. What do I want to happen to the potassium that has leaked out? What do I need to do with that potassium now that it's leaked out? What did I do with potassium? Didn't I pump it in? Okay, so what am I going to do if some potassium leaks out? I'm going to grab it with my pump and pump it back in. Okay? When I do that, what else happens? Don't I also have to exchange that for some sodium? Okay, so every time a little bit potassium leaks out, then you pump it back in, and in the process, that lets you kick even more sodium out. Okay? Until you get to equilibrium. What does it mean to be at equilibrium? It's kind of staying the same. It's not stopping. It's just for every potassium that leaks out, one will get pumped back in. So when a cell is at rest and at equilibrium, then the exchange of sodium and potassium is equalized. And just as many go out as come back in. Okay? Do I have leak channels for sodium? Actually, I might. Okay? And if I do, then what does sodium want to do? If potassium likes to leak out, what does sodium like to do? It likes to leak in. What am I going to do when it leaks in? That's where I'm going to get it to pump back out. So at equilibrium in a cell at rest, a little bit of potassium can leak out, a little bit of sodium can leak in. Not a problem. My pump is just going to keep pumping the stuff that leaks in, back out, and vice versa. Okay, does everybody have a picture of a cell at rest? Pumps are overcoming the effects of the leak channels. All right, so then there's another kind of channel, and our other kind of channel is called a gated channel. So what does a gated channel have? That's a trick question. It has a gate. So when it is appropriately stimulated, then the gate will open in some way. The way these gates work, we've actually been able to picture them. And my favorite one is there's a sodium gated channel that has a gate kind of in the middle rather than on the outside. And the way this gate has been pictured as working is kind of like when you're trying to get in the middle of a row at the theater and you know how everybody sits at the ends because they don't want to get stuck in the middle. So what do you have to do when someone has to walk past you in a theater where the seats are crammed too close together? We're not standing up. Not going to do it. Too lazy. Got my popcorn in my hand. Don't want to stand up. What do? I turn a little bit sideways. I turn my knees sideways. And that's the way these projections in the channel will work. So if you looked at the channel from the top down, what you would see is when it's closed, it looks like this, and that the knees are sticking out straight. And then when it opens, the knees kind of kick to the side. And there's just enough space there for the sodium ions to flow. Okay? To me, it's absolutely incredible that they can image those molecules in those different states and actually see what's going on. But as fascinating as that is, the main thing we need to know is what's going to happen. So if I have a gated channel, and that gated channel, the gate opens. So I'm opening the gate. If that channel would let both sodium and potassium pass through, if it would let both pass through, then what do you think is going to happen first? Okay, I want you to think about this in your groups, and I'm just going to remind you that you have sodium on the outside, potassium on the inside, and you also have these big anions on the inside and some chloride on the outside. So I just opened a sodium-potassium gated channel. Which ion will go through first, or will they go through at the same time? I'm only going to give you about 30 seconds because I just want you to think about it for a sec and then pick. Everybody has to pick a side. Okay, so what factors did you consider before you made your answer? Did I give you all the information you needed? What did I not include in this picture? Well, I have the concentration just in the size of my icons. So I know I have high concentration of potassium and anions on the inside and high concentration of sodium and chloride on the outside. When I first started drawing these, oh, that's as far back as I went. What do you see in this picture that you didn't? These are sloppy pictures. Man. What do you see here that I didn't include here? The channels, the pumps, and what else? What was it? Y'all be brave. Answer loud enough for me to hear you. I have chloride. Yeah, well, that's true. You are correct. But what I'm asking is, what's in this picture that's not in the other picture? The positive and negative charges. So if I add those in here, what do they look like? Positive on the outside, negative on the inside. Does that matter? It does to me. So it does to you. So what difference does that make? Positive would rather go in or out? Positive would rather go in, wouldn't it? Because the inside's negative, the outside's already positive. We talked about that when we talked about the leak channels. So if I open a gate that could let either sodium or potassium go, gate doesn't care, then who's going to push their way through first? The sodium, because it has a stronger drive pulling it in than the potassium has pulling it out. Potassium's already okay, it's very happy where it is with all those anions on the inside. So when I open these gates that will allow either ion to flow through, even though we know both can flow, what we find is that preferentially sodium will come in. Now, what does that do to the charge on the inside? It makes it more positive, right? which don't worry about that because, first of all, these ions can flow. And second of all, I got my pumps running. So they're going to take care of that for me. Okay, so never mind that it will become positive on the inside. We're going to look at that. But for now, that's not an issue. The issue to me is right in here. When I open a gate, ions flow quickly. When I work a carrier like the sodium-potassium pump, that's really clunky and slow. Okay? So when ions are moving quickly through gated channels, they move fast enough to actually create electrical current. So those ions flowing through the gated channel flow fast enough to create electrical current. That's the key. Okay? So, all that being said, how is a neuron like a battery? Does it have opposite ends? Yes. Does it have a switch? What's the switch? The gate is the switch. Okay, what's the wire? The wire is the part that lets the current flow. So it's the channel, the channels. We're not going to work with just one. We're going to open a bunch of these. Okay? So all that's left is, what's the light bulb? And the answer to that is it depends. It depends on what that cell is doing. So if you're at the end of a neuron, you know it's going to release neurotransmitter. That's the light bulb effect. That's the work that's being done. It's going to open some vesicles and release neurotransmitter. If it is the beginning of a neuron, then it's going to carry a nerve impulse. That's the work. Okay, if it's a muscle, we already know it's going to contract. Okay? So let's review our parts. How did we get positive and negative ends? We have positive and negative charges on the outside and the inside. How did we do that? Who gave us that? What part of the cell gives us that positive and negative difference? Is it a pump or a channel that does that for us? It's the pump. So sodium-potassium pumps are continually recharging the battery. This is a living battery, so you don't have to wait and plug it in and charge it later. It charges itself continuously. And it keeps that difference in charge pretty much all the time. Okay, and what was the wire? What carries the electrical current? The channel lets the ions flow. Ions flowing is electrical current on the move. That's electrical current. And what was the switch? The gate is the switch. That one's usually pretty obvious to everybody. Okay, so we have this resting membrane potential. We've got our setup. We know what it looks like. So we understand the role of leak channels, exchange pump, and gated channels. And at rest, of course, the gates are closed. The switch is closed. If I want it to do some work, then I open the gates, and that opens the switch. Is everybody pretty clear on our set point, our resting potential? The abbreviation for this is RMP, resting membrane potential. And if I ask you the value of the resting membrane potential, or RMP, you're going to tell me negative 70 millivolts. Okay? All right, so here's the diagram out of your book. Let's see what we know. What did we call this guy right here? There's our voltmeter. And I have a little plate electrode here and a needle electrode here. I see what looks like a potassium channel. I see my sodium-potassium pump, potassium channel, sodium channel. Okay? This is color-coded for your convenience. So the potassium ions are colored the same blue as the potassium channel, and likewise the sodium it looks kind of purple. And do you see inorganic phosphate, PI? is the same thing as PO4, phosphate. Inorganic, the I just means inorganic phosphate. Okay, any questions about how to read that diagram? All right, so now we're going to talk about what happens when we open some gates. And what we're going to find is it depends. It depends on what kind of gate you're talking about. So we need to talk about our different kinds of gates. And we already met some of our voltage gates when we talked about muscle. So what opens a voltage-gated channel? This is a trick question. Voltage. So electrical current, which is what we measure in voltage, opens the voltage gates. So how did we create electrical current? That's what we just covered. Open some gates, you get electrical current. That electrical current does what? It opens more gates. It opens voltage gates. And that's the work that can be done with voltage. Questions at this point? Okay. Let's talk a little bit about our stimulus-regulated gates because we really haven't talked about those much. We talked about the functional categories, but not the stimulus type. So we have mechanical gates, we have thermal gates, we have chemical gates. All right, so trick question time again. What opens a chemically regulated gated channel? A chemical of some kind. So when neurotransmitter binds to its receptor at the neuromuscular junction, which we just did last week, the acetylcholine binds and the little gate opens. That's a chemical gate. And that's just basically what it means, okay? So if a neurotransmitter can bind to and open a gate directly, that is a chemically regulated gate. And acetylcholine at neuromuscular junction is the perfect example of that because we just studied that. Okay, there are other neurotransmitters that can do that. We also learned last week that for smooth muscle, sometimes hormones can make the muscle contract. Guess what they can do? They can bind to chemically gated channels and stimulate the cell to contract. Okay, so there are other chemicals that it can open gate. When you talk about sensory neurons, are there any sensory neurons that are sensitive to chemicals? Any in your taste buds? Can you sense sugar or salt or acid? Yeah. So how are they working? Those are chemicals that are stimulating neurons and causing gates to open. Okay? You have chemical receptors in your bloodstream. They're very sensitive to pH levels. There are some particular spots. There's one right at the base of your jaw that monitors the chemistry of blood to the brain. There's one coming right off your aorta that measures the chemistry of blood to the whole body. You have receptors in your lungs. You have receptors in your stomach. So you have lots of chemical receptors. When we worry about the ones that have to do with the body systems we'll study next semester, well, we'll do them later. Okay? But this week we're going to look in general at chemical receptors. And the bottom line is there's a little receptor site physically attached to the gate. What happens when the chemical attaches to its receptor site? The gate opens. And as soon as that chemical is removed, the gate closes. So it's really that clear cut. Are you with me on chemical gates? Because they're really pretty simple. Questions about those? Okay? Our second kind I want to talk about is the mechanical gates. Mechanical gates are pretty straightforward as well. Some of your mechanical gates are responsible for your tactile senses, but some of them are also found in your ear, where sound waves cause vibrations, in your stomach and urinary bladder, where stretching of the wall causes mechanical stress. So if vibrations or pushing or prodding or pulling or poking or stretching, if those actually can cause gates to open, those are mechanoreceptors. Those are mechanical gates. So everybody see how straightforward that is? Just stretch the cell and a gate pops open, that's a mechanical receptor. Some of them are a little bit more elaborate. One of my favorites is one that's found in the ear. So in the membranes of your ear, in the membranes of the mechanoreceptors of your ear, here's your phospholipid bilayer. here is the gate, or that's the channel. Here's the gate. And the gate you see is closing it, but that gate is hooked by this little elastic-like protein to this other protein. And this is called a tip protein. And when it pulls, it pops that gate right open. So you just have this like a little spring coil attached to the gate, and it just yanks on it and makes the gate open when that membrane gets stretched from vibrations passing through your ear. So mechanoreceptors, what do they respond to? Mechanical stimulation, touch, pressure, vibration, stretch. Those are the things that cause mechanical stimulation. Some things are not that straightforward. Thermal receptors, we're not really sure how they work. What does it mean to be a thermal receptor? Heat. Might be heat, or the opposite of that could be cold. So we're not really sure how the temperature differences cause the gates to open or close, but we know that they're also chemoreceptors. That is, they're sensitive to chemicals. So when you taste mint, what's the sensation you get? It cools your mouth. It's somehow stimulating the receptors that are the exact same receptors that are stimulated when you drink cold water. And what about hot peppers? Why do we call them that? They stimulate pain nerve endings as well, but they do stimulate heat receptors. And same with other chemicals that feel warm. A lot of the joint creams that people use, icy hot, that kind of thing. It's got two chemicals. One hits the cold receptors, the other one hits the warm receptors. Okay, so we know that our thermoreceptors are kind of a hybrid. They are chemoreceptors because they will open with chemical application, but we don't know really how the thermal effect works. Okay, so I want you to notice as well, though, that these, oh, I forgot to hit my thing. Now I hit it too late. So the ones that we call the stimulus gated, there's two kinds, mechanical, thermal, and chemical can be for our senses. And those, remember, are our unipolar, and in the case of the nose, our bipolars. Okay? But if we're responding to neurotransmitters specifically, then it has to be a multipolar. Okay? Let me put this another way. If I tell you it's a chemical gate, can you tell from that information whether it's sensory or motor? If all I'm telling you is there's a neuron that has a chemical gate, could it be sensory? Yeah, your sense of smell, your sense of taste, could be sensory. Could it be motor? Yeah, because you have neurotransmitters that open chemical gates. Okay, so chemical belongs in both categories. Some sensory neurons have chemical gates, but all multipolar neurons respond only at synapses, which means they respond only to neurotransmitter, which means their stimulus gates are always chemical. Okay? So everybody got that idea sinking in? So where am I going to find mechanical gates? Sensory only. Okay? Sense of hearing and balance, tactile senses, blood pressure, that's a mechanical stress. Chemical is the only one that fits in both categories. Okay? Secondly, where do I find voltage gates? Voltage gates are found only on the part of the neuron that is transmitting the nerve impulse. We're going to stop in just a second and draw a picture because y'all haven't learned these parts yet. And then we're going to come back to this slide again and get the rest of these situations. All right, so I'm going to draw two neurons, and first thing I'm going to ask you to do is to tell me what kind they are. Okay, what's this one, the top one? That's a unipolar or pseudo-unipolar. And what about the bottom one? That's a multipolar because it has lots of cell processes attached to the cell body. Okay? So let's learn some parts. On the unipolar, the part that carries the nerve impulse is called the axon. And guess what that part is on the multipolar neuron? Same thing, that's the axon. This is a review for some of you, I know. These little guys that attach to the cell body on a multipolar are called dendrites. Dendro means branch, so dendrite means little branch. So I have lots of dendrites. These dendrites can have branches on them. So you can have branches on the branches on the branches. How much surface area do I have here? A bunch of dendritic surface area. Okay, so far. The dendrite is the part that stimulates the cell. When I get a stimulus, that stimulus comes to the dendrites. The axon is the part that carries the impulse towards the next destination. So just to make it a simple thing to remember, we say dendrites carry the impulse toward the cell body, and axons carry the information away from the cell body. Are you with me on that? Questions? Okay. So if I'm imagining a multipolar neuron, the cell body may be in my spinal cord. The axon may run all the way to a muscle in my index finger, and when that muscle contracts, it can flex my finger. Okay, so where are the dendrites? They're in the spinal cord. Okay, and the axon runs straight from the spinal cord out all the way to that muscle. One continuous axon all the way to that muscle. When it gets to the muscle, we already studied muscles, so we know that when it gets there, it has lots of branches. And each one of those branches has branches, and each one of those ends in a synaptic knob. Right? Okay. Does a unipolar neuron have dendrites? Very brave of you to guess. Yes, it does, way out in your skin. But look what these dendrites do. They attach directly to the axon. The impulse is carried by the axon this way. Yes, it does stimulate the cell. But the main thing is it's going past the cell body, and when it gets to its end, it's going to do something similar to what the multipolar neuron did. Here it is at its end. It's going to branch, and each one of these branches is going to end in a little synaptic knob. So the problem we have with our very rough sketch of the unipolar neurons, it's hard for you to tell one end from the other. They look a lot alike. Once we get them put in place, it's not hard at all. But synaptic knobs are found only at the terminal end. Okay? They're not found on dendrites. Are you with me on that? All right, so if it's sensory, remember these are unipolar guys, so they're sensory. Where are the dendrites? They're going to be like in your skin. So I have pain receptors and tactile receptors in my right index finger. So in the skin of my index finger, I've got some dendrites. The sensory nerve endings either are the dendrites or are connected to the dendrites. And that, by the way, folks, is scheduled in your online lab to go through all of the sensory nerve endings of the skin. Okay, but I've got some here. Where's the axon? It has to run all the way to the spinal cord. And when it gets almost all the way to the spinal cord, that's where the nerve cell body is. And then the rest of the axon runs into the body of the spinal cord itself and makes connections there. That's where its little synaptic knobs will be. Are you with me on Okay? So we're going to apply some terminology that may or may not help you right now, but once you get this put in place when you go to lab tomorrow, it's going to help you a lot. So the spinal cord, the axon from here to here is almost completely in the central part. It's right up against the spinal cord. Okay, so that's the central segment. So the part of the axon that runs out to your finger, you can call peripheral. You can also call them proximal and distal. Which one would be which? Central is proximal, peripheral is distal. Make sense to everybody? So if you're just trying to talk about the two parts of the axon, that axon doesn't fundamentally change. It's just you've got a benchmark there of where the cell body is. So that's the difference between the two segments of the axon. But to the axon, it's just all one big thing. Okay, so our nerve impulse always starts at the dendrites. And if we get enough voltage to open our voltage gates, then our axons will carry a nerve impulse. Can you picture that, more or less? Okay, so let's review what we know. If we're talking about an axon, what kind of gates do I have? Voltage regulated gates. What opens those? Voltage does. Okay, what kind of voltage gates have we already met? Sodium, potassium, and calcium, and we're going to use the same gates in the same place we did last week. When we look at the parts of an axon, we have the beginning. That's the start of it. It can have branches. Synaptic bulbs, we met, right? Telodendria is a word root that should make at least half sense to you. What did I tell you dendry meant? Same word root as dendrite. It's little branches. But telo, T-E-L-O, is the same word root as in telophase, which was the last stage of mitosis, right? So telo means end. So where do you look for branches? What are you going to call the branches at the end? That's the telodendria. Okay so far? All right, we need to discuss these last points. And in order to do that completely, I think what I want to do next is look at some pictures and make some applications. Okay, so this diagram is showing us some of our gates and pumps. So here is our sodium-potassium pump. And we've already met that. We know how that works, right? Right? So which way does sodium go? Oi. Okay. Sodium gets pumped out. The pump pumps it out. Okay, sodium out, potassium in. Do y'all want to just chant that about 5 times? Sodium out, potassium in. That's why I keep asking. Every semester it's the same thing. Okay. But I'm always afraid y'all are going to get on the job and say, you know, I always got those backwards. And that does not inspire confidence in you as a healthcare professional. So, all right, what kind of gate do we see here? We see a little chemical that when it binds causes the gate to open. So there's our chemically regulated gate. Okay? Here we see our voltage gates at the bottom. And if you can picture what's happening, you see it positive on the outside, negative on the inside. The gate opens. What happens to our charges? They actually flip-flop. And then the gate closes. And we'll talk some more about that. All right, tell me what kind of neuron this is. This is, in fact, a multipolar neuron. So where are the dendrites? Are they at the top of the picture or the bottom of the picture? Dendrites are at the top. And what are these little branches at the bottom? Those are our telodendria. And then the little bulbous expansions at the end are the synaptic bulbs. Okay? So what we need to do is make sure you know where to find each kind of gate. Okay? This is a multipolar neuron. So you've got some chemically regulated gates, and you have some voltage regulated gates. So let's start with the easy one. Where are the voltage regulated gates? Let's go back and take a look. Where are voltage regulated gates? Which side of the list are they on? Are they on the left side or the right side? They're on the right side. What does it say for location, for voltage regulated gates? Axon. Huh? Axon. Axon only. So if I am looking at a dendrite, am I going to see voltage regulated gates? No. If I'm looking at the cell body, the membrane of the cell body might have some gates on it. Are they going to be voltage gates? No. Only on the axon. But the axon is everything from there down. All of it. So the axon starts at this part called the axon hillock. A hillock is a little hill. So it's just a little swelling on the cell body, which is the beginning of the axon. The axon here is myelinated. It's running in the middle of that myelin sheath. There are no branches shown on this one, but it could branch. And a branch would be a collateral. Still part of the axon. What kind of gates? Voltage gates. When I get to my synaptic knobs, is that part of the axon? Yes. What kind of gates? Voltage. Okay? So really, for a multipolar neuron, I only have two choices. I'm going to have chemical gates, which are going to be at synapses on the dendrite and potentially on the cell body. And once I hit my axon, it's voltage gates the rest of the way. And do we remember what opens voltage gates? That would be voltage, which is the electrical current. And what opens are chemical gates? Chemicals, yes, but I'm telling you we're at synapses. So what would you call the chemical that at a synapse will open a chemical gate? That's a neurotransmitter. Very good. Okay, so far? All right, so there's some other issues here that we need to review, but I want you to have this idea in your heads first, this picture of chemical gates at the dendrites and cell body of a multipolar neuron. Once you hit the axon, voltage gates all the way. And before we move off of that, we really should consider what happens with our unipolar guys, our sensory guys. So if we're just talking about a tactile unipolar neuron, that the sensory nerve endings are in the skin, can you tell from that picture where the dendrites are? Are they at the top of the picture or the bottom of the picture? How do you know? Because there's no knobs on the top. So we can recognize their synaptic knobs. This is kind of a general rule, but it's mostly helpful when you're looking at my drawings to be able to interpret them. Okay, so where would I have stimulus-regulated gates? What kind of gates do I have on the axon? Voltage, and only voltage. So where does my axon start? It's axon all the way. So it's voltage gates that whole way. Where's the only place to have stimulus-regulated gates then? Right here. So if I can get back to that page. Here we go. So if it's tactile receptors, the stimulus is mechanical, and just poking it, prodding it, or stretching it may cause the gates to open. If it's a chemical gate, and you have pain receptors in your skin that respond to chemicals being released by damaged cells, That's a chemical receptor, or if it's on the surface of your tongue or in your olfactory epithelium or wherever. Okay? So if it's on the dendrites, it's a stimulus-regulated gated channel. All right? Mechanical, thermal, chemical. All right? So this is really easy. You just got to keep it straight. Of course, I thought the same about the sodium potassium pump, too. Don't get this backwards. What kind of gate on a dendrite? Stimulus regulated. What kind of gate on an axon? Voltage regulated. Okay, just keep that straight. It'll keep you out of trouble a lot. All right, next item on our list there, a graded response versus an all or none. Okay, this to me is like you have two kinds of light switches. Okay, the light switches in this room are all or none. What does that mean? It's either on or off. Voltage gates are like switches. They're either on or off. Once you create enough electrical current to open the voltage gates, that voltage is going to open every switch along the way. It's always going to be exactly enough voltage to open the next gate. Are you with me on the concept? Okay, so what would graded response be like? It's a dimmer switch, exactly. So I can have a stronger stimulus or a weaker stimulus. Can you, for example, tell the difference between warm water and hot water? Can you feel the difference? How do you do that? The more heat that's being applied to your skin, the more you stimulate the dendrites of your thermoreceptors. And the more you stimulate them, the stronger their response will be. So when we call something a graded response, it's kind of like your grade's either an A, B, C, D, or worse. So likewise, you can have a strong response or you can have a weak response, weak or strong. That's the terms you should associate with graded responses. Okay? So if there's not enough salt in your soup, then you're getting a weak stimulus. You add too much salt, then you're getting a very strong stimulus. And it all relates to how much salt there was, how much chemical is being secreted or applied. That determines how many of those gates will open and how long they will stay open. Okay, so imagine for your stimulus-regulated gates that the more stimulus you apply, there are two ways you can get a stronger response. You can either open more gates or you can keep them open for a longer period of time. Is everybody okay with those two ideas? We're going to come back to that. Questions at this point? Okay, does everybody understand the difference between all or none and graded? Got it? Good? Okay. The last point up there is local versus propagation, and this is actually how we give these potentials their names. They're called local potentials if they only apply to the local area. They're called action potentials if they can travel away from the local area. We're going to see how that happens, but the term we use for this traveling effective action potentials is that they propagate. What does it mean to propagate something? You create a new copy of it. You recreate the conditions that caused it to happen. Okay, so a local potential is graded on dendrites from stimulus-gated channels. An action potential will propagate. That is, it will travel from the beginning of the axon all the way to the end. But it's always the same strength, all or none. Questions at this point? This is a really important list. And if you can keep these straight along with the pictures that we drew of the multipolar and the unipolar, so that you know where to look for local potentials and where to look for action potentials, that's going to help you keep it straight. Okay, so when propagation occurs, what does that look like? What was propagation? The traveling of the action potential. So it's going to start up here at the axon hillock, and then it's going to travel, because electrical current can travel, and it's going to travel equally down every single branch to all of the synaptic knobs equally. And then what's going to happen? What? No, you're right. What did you say? Say it louder. The voltage gate. So we're at the synapse. What happened when the electrical current hit the synapses? We had some voltage gates there for calcium. Do y'all remember that from last week? So what happened when calcium came in? It triggers exocytosis, and all those little nerve endings are going to secrete neurotransmitter. And that's the end of the story for this neuron, but it's the beginning of the story for the next cell. So imagine I had a synapse right here, and it secreted some neurotransmitter right on that little dendrite, right up here. What happens on the receiving dendrite? Here sits my dendrite. Here's the synaptic bulb. It secretes neurotransmitter. What kind of gates? Chemical gates. And depending on how much neurotransmitter I secreted, more or less of the chemical gates will open. Are you with me on that? And that then is the new stimulus for the next neuron. So we just go in a series, one neuron to the next. Okay, so far? Any questions about the parts of the gate? Or the types of potentials? By the way, we use the term potential, membrane potential. Potential refers to electrical potential in this case. So it's that separation of charge that gives you the potential. Okay, so we have three kinds of potential. What are they? Three names, three types of potentials that we've just looked at. We had local, we had action, and we had resting. Okay, so those are our three membrane potentials. A resting membrane potential, RMP, -70, you're at your resting values, potassium in, sodium out. Right? Did I say it right? And then if I wanted to create a local potential, what would I do? What do I have to do to start a local potential? I got to open some gates. Okay, that's our switch, right? So I'm going to open some gates. If I'm opening gates to create a local potential, what kind of gates might they be? Stimulus regulated. And what kind of stimuli might I have? Could be thermal, could be chemical, could be mechanical. That's the big three. Okay. In your eye, as we'll see when we get to vision, light energy causes the tripping of some gates. Actually, it causes a chemical change to open some gates. Very complicated, and we're going to skip that story when we get there. Too much information. But you've got the idea. You have a local stimulus that opens gates locally. Now, the next hitch for this, let me see what's next on my list, is that not all local gates are going to stimulate. So let's get through this and then we'll see. Local potentials are decremental. What does decremental mean? It means they decrease. Best example I can give you of this is sound waves. Okay? Are sound waves decremental? Yeah, what happens when you're farther away from the source of sound? You can't hear it. The sound gets fainter and fainter and fainter with distance. So likewise, local potentials are decremental because the signal fades. And the longest distance that can carry effectively is about 1 millimeter away. And guess what the distance is from the farthest tip on the longest dendrite to the trigger zone? It's at most 1 millimeter. Because any further than that, and the signal wouldn't get through. Okay? So when a local potential is created out on a nerve ending, the electrical current created fades with distance. But if it gets through, if there's not competing signals from other dendrites and it gets through to the axon hillock, it's going to excite it and perhaps start a new action potential. Okay, how is that different from action potentials? They propagate. What does that mean? They travel. So even though each action potential at one little spot of the axon doesn't need to carry further than a millimeter, it starts a new action potential at the next little spot. So it propagates by re-upping the voltage. It opens new voltage gates and reboosts the signals, like having signal boosters all the way down the axon. So everybody see the difference between being decremental and propagating. That's the difference between local and action.
So the action potential is kind of like a domino effect?
Yeah, it's kind of like knocking dominoes over. Okay, here is the next big fundamental idea that is different from local potentials and action potentials. And it is that you have two choices. They can be excitatory or they can be inhibitory. Now, we talked about this a little bit last week in regards to smooth muscle. We said that for skeletal muscle, you always start a contraction, right? But for smooth muscle, you actually can produce an inhibitory signal. Do y'all remember that we did that? Okay, maybe not. So the best example of that for smooth muscle is during pregnancy. We know already that smooth muscle, when it stretches, tends to contract. Y'all remember that? So what do you think's happened to that uterus with that rapidly growing fetus in it? It stretches the uterus, and you've got to calm the uterus. Keep it calm. Okay? So you actually secrete hormones that inhibit uterine contractions. They block any signals that would make it contract. Does anybody know what hormone does that? What's the hormone of pregnancy that calms the smooth muscle? It is progesterone. What does the name progesterone tell you? Pro means advocate of. Gester means gestation. So progesterone literally means the steroid hormone for gestation. Maintains the pregnancy by preventing uterine contractions. Okay? Now, at the end of pregnancy, about the 8th month, what happens to progesterone levels? They start to die down, start having contractions. Okay? All right, so we know from last week, or we know from what I said just now, one way or the other, we know that you can stimulate or inhibit smooth muscle. Well, what I'm telling you now is we can stimulate or inhibit neurons, and we're going to look at how we do that, because we're going to see that this has an effect on particular drugs and particular conditions of the brain. So this is important. First of all, we have two kinds of receptors. We have direct and we have indirect. So what do you think it means to be direct? You can read it up there. So if this is my little channel, and here sits my gate, and right here on the channel is the receptor portion that the neurotransmitter is going to bind to, and when it binds, that's going to open the gate. That's a direct neurotransmitter. Acetylcholine is an example of that. Okay? If it's indirect, what we're going to see is that here is the receptor, and over here, in another spot in the membrane, here's my gate. So that when the neurotransmitter binds to its receptor, the binding of the neurotransmitter to the receptor triggers changes inside the cell, and those changes cause the gate to open. That's indirect. I can't expect more explicit terms than that. It either directly binds or it indirectly binds. It's going to open the gate or not. For the most of the neurotransmitters that you're assigned to learn, you do not need to know that. Okay, there are only a few that I'm going to ask you to remember that on. And we already know acetylcholine. Which one is it?
Direct.
It's direct. Okay? All right, now the second part is it's either excitatory or it's inhibitory. This is important. Excitatory depolarizes, that gives you a nerve impulse. Can you figure out what inhibitory means? It means it's going to damp down on its likelihood of sending a nerve impulse. Okay? The terms here are important. Depolarize and hyperpolarize. The ions and directions are important. Sodium and calcium move in to excite. Potassium moves out, or chloride moves in to inhibit. That is an important thing for you to remember. Can you do that? Let's look at a picture. All right, so back to this picture again. This is just a cross-section through, could be a dendrite, could be an axon, doesn't matter. What do I already know? Where's my potassium? Inside. Where's my sodium? Outside. Where's my calcium? Outside. Where's my chloride? My anions are on the inside. I don't need to draw them now because they don't have any gates. They're not going anywhere. These are the 4 ions that we know of that have gates that your neurons can regulate. Are you with me on that? Just 4. All right, so I'm going to need a potassium gate, a sodium gate, a chloride gate, and a potassium gate, or a calcium gate. And we're just going to look at what happens if I open each gate in turn. This is not going to be hard. It's very straightforward. So if I open a potassium gate, what happens? Potassium diffuses which direction? Why does it go out? It's following its diffusion gradient. Okay, we studied diffusion. We get that. Okay? What about sodium? It's on the outside, so it wants to diffuse in. What about chloride? What about calcium? So can we remember potassium is the opposite in this regard to all the other 3? Potassium is the only one that's high on the inside, so it's the only one that wants to diffuse out. The other 3 are on the outside. They want to diffuse in. Okay? Now, what we have to look at next is what will happen for each one when they diffuse. So if we just look at potassium, we know that the cell is positive on the outside and negative on the inside. So what if I open potassium gates and potassium diffuses out? What's going to happen to my electrical charge? I'm going to tell you it will do this. It will. But what happens to the electrical charge? Doesn't it make it more positive on the outside and less negative on the, or excuse me, more negative on the inside because I'm moving positives out. Does everybody see that? Okay, let's graph it. All right, so on my graph, I'm just measuring over time. And I'm measuring the millivolts of my membrane potential. Where do I start? I'm at rest. Negative 70. Negative 70 millivolts. Here I sit, I'm at resting membrane potential, negative 70. I just opened a potassium gate. Not just one, but a few. Which way does my voltage go? Remember, I'm measuring the inside of the cell, not the extracellular fluid. So positive charges are leaving the cell. Doesn't that make the inside more negative? Which way does my line go? Down. These are local gates. They're just transiently opened and closed. As soon as those gates close back up again, I'm going to come back up to rest. A combination of ions diffusing and my little sodium-potassium pumps is going to quickly return me to rest. Okay? Got it? What was the term for this? Okay? Looking back at our picture, is there anything else that would hyperpolarize the cell besides potassium leaving? Remember, to hyperpolarize, we had to make the inside more negative. The chloride, because it's negative and comes in, has the same effect as potassium, which is positive leaving. Does everybody see how that works? So whether it's potassium going out or chloride coming in, either way, that hyperpolarizes the cell. And we say that inhibits the cell. We'll see why in just a sec. Okay? Now that we've seen that, what is the effect of sodium or calcium gates? They're both positive on the outside, so they want to come in. Doesn't that make the inside more positive? So what will that look like on our graph? So if we're sitting at rest at -70, and I open a sodium channel or a calcium channel, which way does my graph go? Up. And again, these little local gates open and then close and get us back to rest. Okay, and what did we call this? Depolarization. Some books will call it a hypopolarization, which is fine because that's the opposite of hyper. I kind of prefer that term, but I don't think your book uses it that way. Okay, does everybody see what the graphs indicate? The change in the electrical potential of the cell itself based on whether it has more positives inside the cell or outside the cell. That's really all it is. Okay, so we need a little bit more background on the terminology. The resting cell is polarized. At -70 millivolts. Why do we say it's polarized? What are its poles? Negative inside, positive outside. That's the polarity of the cell at rest. Okay, so resting cell is polarized at -70. If I make it more polarized, that's hyperpolarized. If I make it less polarized, that's depolarized. What would it look like if I reversed the polarity? Exactly. Positive on the inside, negative on the outside. How would I graph that? Again, over time. So if I'm resting at -70, what would it look like? It's gonna, you have to mark where 0 is, and it's actually gonna go above 0. Okay? Typically, a cell in the middle of its action potential reverses its polarity and goes up to +30 or 35. And then it drops right back down again. Okay, so does everybody get the concepts of the terms of polarity, polarized, depolarized, hyperpolarized, reversal? Got it? Can you graph them? Could you recognize them on a graph? Okay, good. Let's see where we are. I think I'm just going to leave this diagram for you to study in your book. It's a great diagram, but what are we showing here? What do you see in this picture? I see some synaptic knobs secreting some neurotransmitter, binding to a chemically gated cation channel, letting some sodium into the cell. What's that going to do? Do you see the graph shows you depolarization right there? Okay? Questions about that? There's another concept in this picture, and it's a concept I think you met in lab, and that's the idea of threshold. What does threshold mean to you? The minimum. Okay? So did this particular stimulus reach threshold? No, it did not. And threshold is shown in that diagram. If you can read it well enough, can you tell me what the value, the numerical value of threshold is according to this graph? Can't see that far? -55, and that's kind of typical. Okay? All right, so what are we doing here? Quick glance at the figure. What do you see? Look at the graph. We hyperpolarized. We did it at synapses because that's the only place you can do this. Okay? So I have a neurotransmitter I'm secreting that when it binds to its receptor, it opens potassium channels, or what was my other choice besides potassium channels? Chloride channels. Could be either one. Okay? And when those channels open, those ions flow in a direction that hyperpolarizes the cell. And now we're going to see the importance of threshold. What happens to the distance between my potential and threshold while I am hyperpolarized? It widens the gap between the potential and the threshold. Are you with me on that? So by moving away from threshold, an inhibitory stimulus reduces the likelihood that the neuron will hit threshold. There are other stimuli coming in at other synapses, and any neuron at any point in time is being both inhibited and stimulated. And it's just the sum total of all that that determines whether it hits threshold or not. What happens when it hits threshold? That's the trigger that opens the first voltage gates that starts the action potential propagating. Threshold is a phenomenon associated with the axon hillock. That's the only place that threshold matters. Okay? So as an example of something that this relates to, you've probably heard of Valium, have you?
Yeah.
I had the same reaction, actually. When I had my wisdom teeth pulled, the dentist gave me Valium. My sister had to drive me to the dentist. We were away at college when I had my wisdom teeth pulled. And so he said, I'm going to give you this Valium. It's going to make you feel fine. When you wake up, you know, yeah, it's going to hurt, but you probably won't care for a little while. And if you just follow directions, you'll be fine. Can you count backwards starting at 20? I have no idea how far I got because I don't remember any of that. I don't remember the trip home, actually. My sister said I was waving at all the cars while she was driving us home. I have no memory of that whatsoever. But I can tell you what Valium does. We actually use Valium for other things. We use it as a tranquilizer, yes. We use it as a drug to end seizures, actually. So what Valium does is it opens chloride channels. It binds to and opens chloride channels. So what's it going to do? Chloride hyperpolarizes. It moves neurons away from threshold. It makes them less excitable, which calms your seizures or puts you to sleep or calms your anxiety or whatever. Okay? Does everybody see how that works? On the other hand, MSG. Have you heard of MSG? Monosodium glutamate? Yeah, it's used a lot in Chinese cooking. So it causes what we call Chinese restaurant syndrome. Heard of that? Please do not try this at home. I've done it. It's weird. But if you have blood pressure problems, I'm afraid it might actually harm you. I was just sitting there going, oh, is this what happens when you get too much glutamate? Glutamate is actually a neurotransmitter in your brain that opens calcium channels. So you have glutamate releasing neurons in your brain, and you have these glutamate pathways. And they all work by opening calcium. What does calcium do to neurons? Does it excite or inhibit? You should have that written in your notes. It excites them, so they're all excited. They're sitting there getting excited. That's great. So you're sitting there having eaten too much MSG. By the way, I did it to myself because I was cooking at home. MSG is the flavor enhancer. You might have it as accent, and you get it in little bottles, and you're supposed to put a little bit on your food. Yeah. Well, we bought it at Sam's, so we had this honking big thing of generic MSG, and I just kind of overdid it. I was making homemade wontons, which are great, by the way. So, yeah, they really tasted good. I still remember that part, but I also remember sitting there later just being like really alert and really unable to focus on a single thing other than, boy, am I really alert, and gee, you know, I'm kind of sweating here. So that's all I remember. So it's not worth experimenting with. It doesn't do you any good. But glutamate is also called the stroke neurotransmitter. This is why I don't want you playing with it at home. When a patient has a stroke, then neurons die, right? And neurons, when they're dead, tend to, some of them anyway, rupture. And if they are in a glutamate pathway, then when they rupture, they release their glutamate, which then stimulates the next neuron, which then takes in calcium. But because it's overstimulated, there's too much glutamate, that much calcium coming into the cell kills the cell. Well, guess what happens next? It ruptures and releases all its glutamate, which then causes the next cell in line to let in too much calcium. So you can see where that's going. So we know there are these radiating damages associated with stroke, and what we found is they're in these glutamate neuronal pathways. So glutamate in excess can actually kill cells by having just such an overload of calcium that it literally causes the cell to die. Okay, so in general, you have these two concepts of excitation and inhibition, and they are going to get integrated at the level of the neuron. So we need to see what happens at an individual synapse, and then from there we'll be ready to integrate. So to start with just some terminology, so you have a presynaptic neuron and a postsynaptic neuron. Can you tell me which one's which? I'm pretty sure we can get those names. Which one secretes the neurotransmitter? Presynaptic's coming in, it gets an action potential, it hits those voltage-regulated calcium gates, that triggers exocytosis of the neurotransmitter into the synaptic cleft, which we met last week. And the receptors for the neurotransmitter, of course, are going to be on the postsynaptic neuron. Right? Good? Okay. We'll see that there are quite a few differences between them. Here is a typical diagram. So what do you see in this picture? There you see calcium voltage gates letting calcium in, calcium triggering exocytosis of neurotransmitter, neurotransmitter binding to its receptor. Is this a direct or an indirect gate? Direct, because the neurotransmitter is binding to its receptor that is part of the gate and opening the gate. Okay, so that's pretty typical for a synapse. So if I use the terms presynaptic and postsynaptic, I expect you to know what I mean. All right, we're going to put this together into a reflex, and you're actually going to study reflex arcs in lab tomorrow, so I'm not going to spend a lot of time on this. I'll leave that for lab. But I want you to focus in on what's happening at our synapses and where our synapses are. So our first synapse is going to be between that sensory neuron and an integration or association neuron, if there is one. And then the second synapse is between the interneuron and the motor neuron. Okay, these are going to be excitatory. They could be inhibitory. At least some of them could be. So when the sensory information comes in from your hand, say you touched a hot object, what are you going to do when you touch a stove that's too hot? We call that a withdrawal reflex. So what happens, and it happens, of course, very rapidly, is the overload from the sensory receptors fire off an impulse which travels at a rate of about 100 meters per second, so it doesn't take very long to get there, right? And then at that first synapse between that and the integrating neuron, the middle guy, that's going to be excitatory. It's going to secrete neurotransmitter. It's probably direct. It's probably going to immediately secrete enough neurotransmitter to fire off an action potential of our second guy, which is purple in our picture. And he has a very short axon, so it doesn't take very long to get to the motor neuron. What's the motor neuron going to do? Again, that's going to be excitatory, and it's going to send signals out to make you contract those muscles. But wait a minute. If I'm doing a withdrawal reflex, are y'all looking? If I'm doing this, what am I doing to these antagonistic muscles? Because I sure don't want them contracting at the same time, right? So I'm going to inhibit them. I'm literally going to have more branches coming off my association neuron that's going to go to the antagonistic muscles neurons and turn them off, inhibit them. Make sense? Likewise, you have what we call a cross-extensor and flexor reflex. So imagine you stepped on a sharp object, which in my household these days is usually Legos. It used to be Barbie shoes, because I only have those. Okay, so if I step on a sharp object, what do I do with the leg that is now sending signals saying damage, danger? I need to flex that one, right? Withdrawal, a flexor reflex. But what do I better do something with my other one. So what am I going to do with that? Extend. Okay, so I'm literally shifting my weight, and you can imagine how complex those two reflexes are in terms of the muscles that I have to contract versus the muscles that I have to inhibit to keep them from contracting. Are you with me on all that? You do this all the time. You're constantly balancing, right? So, and as you balance, the cerebellum of your brain is doing a lot of the work involved in that, but a lot of that work's just in your spinal cord. So you have stretch receptors in your tendons and in your muscles that you're going to learn in lab tomorrow. When a muscle stretches, when I flex my biceps, what am I doing to my triceps? I'm extending it, right? I'm stretching it. There are stretch receptors in there that send signals to my cerebellum saying, hey, I'm stretching. And at the same time, what is the biceps tendon doing? Isn't the biceps is contracting, but because it's pulling on the tendon, isn't the tendon stretching a little bit? So my tendon is sending signals saying, this muscle's contracting. So if a muscle stretches, that means it's being stretched. But if a tendon stretches, that means its muscle is contracting. So if I see that, that's kind of two opposing signals, but the signal is this side's contracting, this side's relaxing, my cerebellum's keeping track of all that, but my spinal cord is as well. So as you walk, you have locomotor patterns that are just wired into your spinal cord. And if you've ever watched a baby learn to walk, you can kind of see it progress as they get better at it. And a lot of that feedback is the frustration of falling over all the time. Mine, the 2-year-old now, is at the point where he's just now beginning to look where objects are at head level. He's so tired of hitting his head. He's finally learning to look. Okay? And for us, you know, that's just second nature. We do it. But it's all learned. It's all feedback. Those locomotor patterns in your spinal cord are hardwired. And the patterns are kind of set so that your cerebellum doesn't have to keep track of that. Once you learn how to walk, you just kind of keep doing it. But they are based on stretch receptors in the muscles and in the tendons, and that impulse is constantly coming in. So you are constantly stimulating the motor neurons in your spinal cord all the time. But you're not sitting there twitching, are you? Pretty sure y'all aren't. I would have noticed. But people do. They have seizures. They have convulsions. Sometimes those are generated in the brain. Sometimes they're generated in the spinal cord. And you have to calm those neurons down because they're constant. Every time you move, you're sending impulses in, saying, I'm moving, I'm moving, I'm moving. So what is the natural reaction if you lean too far this way? This side's getting stretched, this side's bending over. So to balance yourself, what are you going to do? Contract the other muscles that are being stretched to pull back. And that's part of balancing, and all of that's part of that locomotor pattern. But if you didn't keep it calmed down, then you would overcompensate. And so you would go too far, and then what? You would go back, and you would go back. And this is the cause of convulsions. So whether it is tetanus, the disease, or strychnine poisoning, and again, I'm not trying to teach you guys how to do people in, but with strychnine, you inhibit one of the inhibitory neurotransmitters. So if I inhibit the inhibition, what's that going to do? The net effect of that is to excite. And so you get, from strychnine, you get convulsions because you're inhibiting an inhibitory neurotransmitter. From tetanus, the muscles are just twitchy. We'll see why next class. But once you stretch them, they overcompensate. And then that causes the opposite muscle to do the same thing. And before you know it, someone with tetanus is just pulled up into a rigid state, all the muscles rigid. Okay, so how important is it to understand all this stuff? Yeah, we're going to see it's very important. Okay, we are about out of time. Any last minute questions? Let's just take a look at where we got to get by tomorrow. What chapter are we in? Who can answer that question? We're in chapter 12. Yeah, we got a ways to go. We'll finish it tomorrow. Okay, so I'll see you then.